Hello friends, you're watching my channel Biologics 5. I've already started a unit of biotechnology. In my last video, I've discussed how first recombinant DNA is formed. In this video, we will talk about the restriction enzymes. These two restriction enzymes in the cloning vector are the important tools in the field of biotechnology. Restriction enzymes are the indispensable tool we can say in the field of biotechnology and recombinant DNA technology, because without them none of the processes like gene cloning, gene mapping, genetic engineering will be possible. None of these processes can be possible without the help of restriction enzyme. Restriction enzymes are actually the type of nucleases. They are also known as molecular scissors. They are also known as molecular scissors because they cut the DNA in a specific manner. These are type of nucleases. Actually, it is belong to the group of nucleases. Nucleases are the enzymes which cut the DNA in a specific manner. Nucleases are the enzymes which cut DNA, and these are of two types: exonuclease and endonuclease. These are of two types: exonuclease and endonuclease. Nuclease and endonuclease. And restriction enzyme is a type of endonuclease because it cuts the DNA strand in the middle or within the strand. In the middle or within the strand. This is endonuclease. Exonuclease cut the DNA strand from the terminus or from the endpoints. So these are known as exonuclease. Restriction enzyme is a type of endonuclease. Restriction enzymes are the type of endonuclease. They cut the DNA strands in the middle somewhere or at the restriction sites and within the strands. They cut the DNA within the strand. Originally they are discovered in bacteria. That use them as their defense system to restrict the growth of viruses. Viruses also attack on bacteria, and the viruses which attack on bacteria are called bacteriophages. So in 1963, two enzymes responsible for restricting the growth of bacteriophage were discovered or isolated. These two enzymes were important methylase and the restriction enzymes. So restriction enzymes first originally in the bacteria in 1963. Suppose this is our bacterial cell. And this is the genomic DNA of the bacterial cell. And this is the phage attack on bacteria. Viruses attack on bacteria known as bacteriophage. So this is the phage which attack on bacteria and transfer its DNA into the bacterial cell. So here the function of these enzymes started: methylase and restriction endonuclease or restriction enzymes. What is the function of restriction enzymes? It cuts the DNA. So this restriction enzyme of bacteria degrade this DNA of bacteriophage or virus attack on bacteria. And what is the function of methylase? Important enzyme also. So this methylase actually protect this DNA, genomic DNA of bacteria from the action of restriction endonuclease or restriction enzymes, because they are vulnerable to the restriction enzyme. Because restriction enzyme cutting this phage DNA, it can also cut the genomic DNA of bacteria. So these methylase enzyme protect the genomic DNA of the bacteria from the action of restriction enzymes. It methylated the certain nucleotides in the genomic DNA and protect it from the action of restriction enzymes. So these two enzymes, methylase and restriction enzymes, are the defense system of bacteria. Bacteria protects its own DNA through methylation at specific location with the help of methylase. And restriction enzymes cut the DNA of bacteriophage. The first restriction enzyme which is isolated is hinsekin. is the first restriction enzyme isolated and characterized 5 years later. It is isolated first restriction enzymes and characterized after 5 years. Today we more than thousands of restriction enzymes isolated from different strains of bacteria. Most type 2 restriction enzymes. Most type 2 restriction enzymes cut DNA into staggered end, for example. This is given. Why we are saying that type 2 restriction enzymes? Because there are different type of restriction enzymes. Like restriction enzymes are of actually 4 types. First, second, third and fourth. But this is the most commonly used restriction enzyme in the field of biotechnology because of some specifications. Why? Because they produce the staggered end or sticky ends. They cut at the specific, they cut the DNA at specific location within the recognition site. Suppose this is the recognition site of DNA and it cuts within the restriction sites, Some enzymes like first, second, third are actually do not cut the DNA in the recognition sites. They cut the DNA far away from the, sometimes away from the restriction site. They recognize the sequence, but they cut the DNA away from the restriction site and sometimes produce blunt ends or even. So this restriction enzymes, second restriction enzyme, type 2 restriction enzyme, cut within the recognition sequence and produce the staggered end like this, and these are known also known as sticky ends. These are overhanging stretches of DNA produced by the restriction enzyme type 2, and these are sticky ends and can be ligated with other DNA molecule which have complementary bases on them. So type 2 restriction enzymes cut DNA into staggered end and quite useful in the field of biotechnology. Next we come to the nomenclature of restriction enzyme. How restriction enzymes are named and what are the rules behind them. Begin with the 3-letter acronym. First comes from the genus of bacterial cell from where the enzymes were isolated. Suppose we are taking here for example taking some enzymes like ECO R1. Next we are taking him 2. Next we are taking BAM H1. So these are the restriction enzymes and we will discuss how the name of these enzymes appears. The first letter, this is a 3-letter acronym and the first letter comes from the genus. So this is the first letter of each enzyme, that is in capital. This first letter comes from the genus of the bacteria from where these enzymes were isolated. So EcoR1 isolated from Escherichia coli. So we have taken capital E in the form of, which is genus of Escherichia coli is Escherichia, and the capital E comes from the Escherichia. Next 2 letters come from the species. Next 2 letter comes from the species. The species of Escherichia coli is coli. So we have taken 2 letters from coli, CO. And the third letter could be added to indicate the individual strain. So R here is the strain of Escherichia coli. So R is taken from the strain. The Roman numeral, this is the 1, and the Roman numeral 1 comes from the order in which the enzymes were isolated. We can also take EcoRI second, means this enzyme EcoR1 isolated prior to the EcoR2 and it is isolated later. So here we will take R1 which is isolated prior to 2. So this Roman numeral indicate the order in which the enzymes were isolated. Here we are taking example of this EcoR1 isolated from Escherichia coli and the strain is RY13. So the E word, the E letter comes from the genus, and two letters, small letters, come from the species, and the third letter comes from the strain, and this Roman numerals come from the order in which they were isolated. In hint second, taken from the Haemophilus influenza, isolated from Haemophilus. H comes from the genus, IN comes from the species, and this D comes from the strain, and the second Roman number and the last Roman number isolated. Roman number means the order in which the enzymes were isolated. So this is all about the nomenclature of restriction enzymes. Now we come to how restriction enzyme functions. Each restriction endonuclease recognizes specific sequence that is known as palindromic nucleotide sequence of the DNA. This is around 6 to 8 nucleotide long sequence, 6 to 8 nucleotide long sequence, which is recognized by, and it is a palindromic sequence. What do we mean by palindromic sequence? Palindromic means is a sequence of base pair that reads same on two strands when orientation of reading is kept same. For example, here this is the palindromic sequence on DNA strand, and if we read this sequence in the 5 to 3 prime order, in this strand this is the 5 to 3 prime order, and we read the strand GAATTC, and the antiparallel strand is complementary strand. If we read the strand in the same orientation, means 5 to 3 prime. This is 5 to 3 prime of this sequence. So if we read this in 5 to 3 prime direction, it will remain same, GAATTC. In this strand GAATTC in 5 to 3 prime orientation, and here also in 5 to 3 prime orientation GAATTC. So palindromic is a sequence that reads same on two strands when orientation of reading is kept same. Restriction enzymes cut the strands of DNA little away from the center. This is the functioning, how this cuts the DNA strand little away from the center of palindrome, but between the two bases on the opposite strand. Suppose this is the center of our palindrome, and restriction enzymes cuts little away from the center. Here it will cut, but between the two bases of opposite strand. Here this is a palindrome, and restriction enzymes cuts here and produce the staggered end. So restriction enzymes cuts little away from the center of palindrome, but between two bases, between these two bases on the opposite strand, between G and it will cut between the G and A. This leaves, what this leaves? Single strand portions or overhanging stretches, most important sticky ends at ends, which are called sticky ends. So here we can see in the picture clearly here, this is our DNA strand and this is our palindromic sequence on the DNA strand, and this restriction enzyme cuts here between G and A. It will break the phosphodiester bond between these two nucleotides, and here this cuts here between G and A on both strands and produce this staggered end. These are the stagger ends of the DNA produced by the restriction endonuclease. EcoR1, this is the recognition sequence of EcoR1. So these staggered ends are produced with the help of restriction enzymes. These sticky ends which are formed by the action of restriction enzymes can be ligated with the complementary sequences present on other sticky ends of DNA. Other strands of DNA can be ligated to these ends, and in this way recombinant DNA can be formed. They are named so, sticky ends are named so because they form hydrogen bond with their complementary counterpart. So these sticky ends most important in the field of biotechnology, produced by the restriction enzymes. Through these ends we can easily ligate these DNA strands to other DNA strands which have complementary bases against them. So here the steps which involve in the formation of recombinant DNA by action of restriction endonuclease enzyme EcoR1. Here's the picture. We can clearly see here this is the vector DNA which we have to transfer our foreign gene. This is the vector DNA. Vector DNA is the DNA which carry the foreign gene, and then we can transfer this recombinant DNA into the host. So how will we introduce our gene or ligate our foreign gene into this vector DNA? Here the action of restriction endonuclease occurs. First we treat both DNA strands with the same restriction enzymes. Most important point. We treat both strands with the same restriction enzyme, and that is EcoR1. We treat both DNA strands with the same enzyme, so they produce the sticky ends which are complementary to each other. So they produce the sticky ends which are complementary to each other. So in this way the complementary sticky ends are produced with the help of EcoR1, and they can easily ligate it to each other and form the hydrogen bond. With the help of ligase, with the help of enzyme ligase, we ligate these both strands and the recombinant DNA is formed. These are the steps in the formation of recombinant DNA by EcoR1. So in this way EcoR1 play important role in the field of biotechnology and in the formation of recombinant DNA. So friends, this is all for today. If you like my video, please like and share it, and don't forget to subscribe my channel. If you have any queries or any questions regarding this topic, you can write me in comment box. You can also join me on my Telegram channel. I will provide the link in the description box. Thanks all for today.